In the previous section, which I titled Part A, I looked at some rare genetic anomalies that relate to male and female development, which I referred to as genetic surprises. And in this section, Part B, I want to look at some more examples of genetic surprises in humans. So the first condition that I want to look at relates to male development, and this is referred to as persistent Mullerian duct syndrome. Now, to understand this condition, it's important to remember that in the early stages of in utero development, male and female babies possess both Wolffian ducts and Mullerian ducts. Wolffian ducts give rise to male reproductive structures, and Mullerian ducts give rise to female reproductive structures. So standard female development involves getting rid of the Wolffian ducts, and standard male development involves getting rid of the Mullerian ducts. Now, in the case of males, the way this happens is that the embryonic testes not only produces testosterone, it also produces anti-Mullerian hormone. As the name suggests, anti-Mullerian hormone prevents the development of the Mullerian ducts and thus prevents the development of female reproductive structures. However, in the case of persistent Mullerian duct syndrome, mutations in specific genes can actually result in a male with standard XY sex chromosomes who develops male anatomy and reproductive anatomy, but he may also develop a uterus and fallopian tubes. This is how de Groot's endocrinology describes the genetic basis for this condition. Persistent Mullerian duct syndrome is a rare autosomal recessive condition that most commonly results from mutations of the AMH or AMH receptor type 2 genes, though idiopathic forms with no identified molecular cause have also been reported. Now, this condition is incredibly rare. In the study that I'll cite here, there is less than 300 cases that have been cited in the medical literature. But once again, it's interesting to note that with this genetic surprise, mutations are once again the culprit. So what I want to do now, I want to change tact again. I want to talk about a different condition, and this condition has actually become rather contentious in the context of elite sport, because it sometimes prompts people to ask, is that a male participating in a female sporting category? Now, this condition relates to individuals that have XY sex chromosomes and a functioning SRY gene. But to understand this condition, it's important to realize as background information that standard male development isn't just dependent on testosterone, it's also dependent on dihydrotestosterone, which is a more potent form of testosterone and is actually made from testosterone. Now, dihydrotestosterone is critical in forming the external male anatomy. Dihydrotestosterone is formed from testosterone via an enzyme called 5-alpha reductase 2. This enzyme is encoded in the gene SRD5A2. However, if deleterious mutations occur in this gene, someone may not form dihydrotestosterone, which will impair their ability to form external male anatomy. So to conceptualize this, I want to show my pathways diagram again, but once again, I'm conceding that this simple diagram can't convey all the complexities of this condition, but nevertheless, here we see in the pathways diagram, you see that I've got an XY individual. They have a functioning SRY gene, so that lever setting off male development is pulled, and the testes start to develop and so on. But then notice down the pathway of male development, I have put an exclamation mark. Now, the reason I have put that exclamation mark is to show there is a problem with male development. Without the ability to form dihydrotestosterone, this will impair the ability to form male external genitalia. Consequently, a baby can be born where the physical appearance is female. However, when we consider this condition in the context of elite sport, people will contend, well, these individuals still have internal testes and they have cells that can respond, and these testes produce testosterone, and they also have cells that can respond to testosterone, and therefore they will benefit from the muscle strengthening that occurs when males go through puberty. And that's why this condition is so contentious in the context of elite sport. Now, this condition is very rare, but it's hard to find any solid figures on how common it occurs. However, de Groot's endocrinology does say this about where it's more likely to occur. Steroid 5-alpha reductase isoform 2 deficiency is a recessive autosomal disorder that is frequent in communities of Turkey, the Dominican Republic, and Papua New Guinea where consanguinity occurs, but relatively rare in other parts of the world. Now, when they refer to consanguinity, they're referring to close relations having offspring. We all have mutations in our DNA throughout our genome. However, close relatives are likely to have mutations in the same genes. Therefore, when close relatives have offspring, the resulting child is far more likely to be homozygous or have 2 copies of mutant genes and therefore display the mutant phenotype. And that's why this condition, 5-alpha reductase deficiency, is more common in these parts of the world where consanguinity more commonly occurs. So what I'm going to do now is I'm going to change tact again and look at a condition where a baby can be exposed to an excess of a particular sex hormone. But to understand this condition, we need to return to a comment from the endocrine, a statement from the Endocrine Society, which I have previously referred to, where they said, women have testosterone too, but in much smaller amounts than in men. Now, the fact that women have some testosterone raises an important question. And that question is, where do they get it from? If testosterone is produced by the testes and women don't have testes, where do they get testosterone from? Well, the answer to this is that women do get some testosterone. Their ovaries produce some testosterone, and so do their adrenal glands. Now, your adrenal glands are these small glands that sit above your kidneys, and they produce some different hormones, including things like cortisol, but they also do produce some testosterone. However, in a condition known as congenital adrenal hyperplasia, the adrenal glands can produce an inordinate amount of testosterone. And this can have a big impact on the development of a baby girl, for instance. So let's see what the Endocrine Society has to say about this condition. 46XX DSD occurs when the fetus is exposed to excess male hormones at the time that the genitals form. The most common cause is congenital adrenal hyperplasia. In this condition, the adrenal glands, small glands above the kidneys, overproduce androgens, male hormones. So think of it this way. We have a baby girl with XX chromosomes developing, but while they're developing in utero, they are exposed to very high levels of androgens, which are male sex hormones. This would have a tendency to masculinize some of their features. Now, and of course, we're not talking here about taking cross-sex hormones much later in life. They are being exposed to these high levels of androgens while they are developing in the womb. This is how the Endocrine Society describes the genetic basis for this condition. Congenital adrenal hyperplasia, also referred to as CAH, is a genetic disorder in which the 2 adrenal glands do not function properly because of mutations in the gene encoding adrenal steroid 21 hydroxylase, which is an enzyme. Without this enzyme, the adrenal glands may produce too little cortisol and or aldosterone and too much androgen. children can inherit an abnormal copy of the gene from each of their parents. So how common is this condition? Well, according to de Groot's endocrinology, this form of congenital adrenal hyperplasia occurs in about 1 in 14,000 to 1 in 18,000 live XX births. But notice once again, the culprit is a genetic mutation, and of course, the severity of this condition will vary as well. So that's an example where a baby is exposed to an excess of a particular sex hormone. But now I want to look at the flip side of that, where an individual can have a reduced sensitivity to a particular sex hormone. But as background information, I need to go back to a condition that I referred to in Part A, complete androgen insensitivity syndrome. Now, as you'll remember, in this condition, because of mutations in the androgen receptor gene found on the X chromosome, cells are completely insensitive to the effects of male androgens, male sex hormones, and therefore the male developmental pathway is avoided, is bypassed. However, instead of complete androgen insensitivity syndrome, people can have partial androgen insensitivity syndrome. Therefore, their cells will have a reduced capacity to respond to androgens, but they'll still be able to partially respond to them. This would, for instance, if you had an individual with XY sex chromosomes with partial androgen insensitivity syndrome, therefore, androgens would have a reduced capacity to, for instance, masculinize one of those individuals. But of course, the presentation will vary. And once again, the main reason for this is because of mutations in the androgen receptor gene. This is a little bit analogous to type 2 diabetes. In type 2 diabetes, the cells of individuals become resistant or insensitive to insulin. But of course, this insulin resistance will vary from mild to severe. In the same way, in partial androgen insensitivity syndrome, their resistance to androgens could vary from mild to severe, and therefore their impact on their development will be from mild to severe as well. So that sort of ends our very brief tour of some of the main genetic surprises that we encounter in humans that relate to development in males and females. But what I want to do is quickly survey what we've looked at and some of the genetic basis for that. So first of all, we looked at Swyer syndrome, where we had a female with a Y chromosome, but because of mutations or deletions in the SRY gene, the male developmental path was avoided altogether. We looked at complete androgen insensitivity syndrome, and there we saw mutations in the androgen receptor gene prevented cells from responding to androgens, which are male sex hormones. In the case of XXSRY plus males, we saw that because of an abnormal exchange between the X and Y chromosomes, a man could pass on an X chromosome that contained an SRY gene. In the case of persistent Mullerian duct syndrome, we saw that mutations in specific genes prevented the breakdown of the Mullerian ducts, which usually occurs in male development. In the case of 5-alpha reductase deficiency, we saw that mutations in a gene encoding an enzyme prevented the formation of dihydrotestosterone, which impaired the development of the male genitalia. In the case of congenital adrenal hyperplasia, we saw that mutations in a particular gene encoding an enzyme caused the adrenal glands not to function properly. And in the case of partial androgen insensitivity syndrome, we also saw that mutations primarily in the androgen receptor gene impaired the ability of testosterone to perform its function in masculinizing an XY individual. What we have primarily seen is genetic glitches or genetic mutations that have somehow impaired male or female development. That's why these conditions are often referred to as disorders of sexual development or differences of sexual development. Now, I can understand why people are preferring to use the term differences of sexual development instead of disorders of sexual development, because the word disorder carries more negative connotations. But in any case, the way that DSDs come about is primarily through genetic mutations or genetic glitches or chromosomal aberrations. It's not like we throw out our whole understanding of the XY system and go, well, that no longer applies. I would contend it does. It's just that a genetic glitch has somehow made things a bit more complicated. But the fact of the matter is, all humans are impacted by mutations. We all have plenty of glitches in our genome. Now, fortunately, because genes come in pairs, if one of them is corrupted by mutation, we have a backup copy. So a lot of problems are mitigated because of that. But nevertheless, we all have plenty of glitches in our genome. If you were to send a DNA sample into a company that does health checks, they could inform you about your susceptibility to all sorts of diseases because of mutation-induced variation in your DNA. And when we consider something like cancer, cancer is essentially a disease of the genome. As mutations impact the DNA in our cells over time, cancer can result. That's why as we age, our susceptibility to developing cancer increases. So all individuals are impacted by mutations because mutations have a tendency to corrupt genetic software, sometimes in really subtle ways, sometimes in really profound ways. And as we've seen, sometimes people have mutations in parts of their genome that affects their sexual development. However, by focusing on rare genetic exceptions, I think it's easy to get things a little out of perspective. So for instance, if at this point you're starting to think, well, the real difference between males and females genetically is basically just one chromosome, and then again, it's mainly just one gene on one chromosome, it's mainly that SRY gene. If you're starting to think that way, I think you're starting to get things out of perspective, and I'm not doing a very good job of explaining things. So at this point, I want to reintroduce Jenny Graves to give us a genetics reality check. In an interview she conducted several years ago, Jenny Graves said this: "Men and women are biologically very different. It's not just the genes on the Y chromosome or the genes on the X chromosomes. It's 1/3 of the genome is expressed very differently in one tissue or another, or all of them. So they are not equal if you think of equal being biologically the same." Now, what she's referring to here is research published back in 2017, where researchers studied gene expression in males and females in 53 different tissue types from post-mortem samples. And in an article I'll quote now, Jenny Graves discusses what these researchers found. They found that about 1/3 of these genes, more than 6,500, had very different activities in men and women. Some genes were active in men only or women only. Many genes were far more active in one sex or the other. A few of these genes showed sex bias activity in every tissue of the body. More commonly, the difference was seen in one or a few tissues. So you see, instead of thinking, oh, okay, well, there's just really one gene that's separating males and females, perhaps instead you should be focusing on the discovery that males and females are using about 6,500 genes very differently. No wonder Jenny Graves summarized at the end of the article I just cited, "Like it or not, evidence now shows that men and women differ genetically far more profoundly than we have previously recognized." But it even goes deeper than that, because not only are males and females genetically very different, we're also epigenetically different. And this is a profound difference that I want to look at in the next section.